Kashmila, is my screen visible? The PubMed.
Yes, ma'am. Yes.
Okay. Thank you. Thank you. Okay. We'll go to the PubMed, and I am starting from the scratch. And starting from the scratch, the very first thing is I have to identify the disease. I have to identify the target. Okay. Of course, there are literature mining things are also there, but as a beginner, we will not go for the literature mining. We will understand how we can search the literature. So we can go to the advanced search. Of course, many of you must be knowing how you can access this, but I will make it little bit easy for you. If you will type the diabetes term, you will get more than 3 lakh research paper, and all of them are not for use. Okay, so what we will be doing here, we will be typing here the type 2 diabetes. Okay, and target gene. Let's search. We are getting only 4759 results. Okay. So what you have to do, like here, we have to go to the research article and we can read a little bit about it and we can understand what exactly the latest gene people are studying. Okay. What are the genes, what are the proteins people are working on, and what we can do, we can work on that particular protein itself. Okay, that is the first thing. From here, we can get the literature is the most important thing for any researcher. If you have to do research in any area other than CAD also, you have to explore the literature definitely. Okay, that is what your PubMed is. Then comes this PubChem, Unipro, Drug Bank. These are the CAD related resources basically. Okay, PubChem and Drug Bank are the repositories for the compounds, chemical compounds. Okay, Unipro, PDB are the repositories for the protein. Okay, and cadd is the repository for the pathways. Okay, that is the deep understanding. Pathways, like for example, I am selecting the gene SERT1. Now next I want to see in which pathways the SERT1 gene is involved in. Okay, while targeting any target gene, any protein, we have to make sure that by targeting that particular gene, we are not affecting any other process, human normal process in the human body or any other organism you are considering. Okay, so these are additional analysis we have to do. So let's move to the another database, that is the PDB. Okay, that is the homepage of the PDB. Here you have to enter your protein, whatever the protein you want to look for. Okay, for example, I want a neuraminidase, I will check. Okay, so while checking the protein, you can type your protein, and here are the refinements on the basis of the structural like the what which method has been used to determine the structure. Okay, the scientific name of the source, like organism, which organism you want to look for. Like here, the only influenza A is there, so there is no need to further selection. According to the taxonomy, according to the experimental method, as we know, the X-ray diffraction and NMR2 method basically used for the crystal structure of the protein. So here you want to, like here only one things are there. There are no many things, but if you will type some, for example, I will type the more B. Okay. So here, can you see? It is present in multiple organisms. Which organism you want to work on, just select that organism. Which taxonomy, whether the bacteria, archaea, or any other unclassified segment, you just make it. Whatever the refinement you will be doing here, that will be done. Now